This is part 6 of the chemistry lecture, and this should be the last. This should be the last one. Okay, so proteins can denature, and when they do, that can lead to illness. Excuse me, it can lead to illness, and it can also lead to death. So denaturation is a change in the three-dimensional shape of the protein, or the tertiary shape of the protein. And it is caused by changes in temperature, changes in pH, or the ionic composition of the surroundings. In other words, the concentration of sodium or chloride or potassium, that kind of thing. Denaturation of proteins can cause irreparable damage to organs and organ systems with loss of structural proteins and enzymes. Sorry, I'm pretty sure I had my pen out and it's not charged anymore, so I can use my finger because it's a touch screen. So enzyme function, we've talked about enzymes, that what they do is lower the activation energy of chemical reactions so that chemical reactions can happen faster. And this is basically how they work. This is the enzyme. Let me try this again. If this is the enzyme, notice how it has two spots called active sites, and they kind of match up with the shape of two areas on the substrates or reactants. So what's happening with this enzyme is it is helping to combine substrate 1 and substrate 2 into a product. So they're binding with the active site of the enzyme, creating a product. The enzyme is then releasing that product. So an example of what this could be is S1 could be glucose and S2 could be fructose. And then the product could be sucrose. But the enzyme can be used over and over again to link glucose and fructose to create sucrose. The enzyme comes away from the reaction unchanged, and that same enzyme can be used over and over and over again as long as it doesn't get denatured. Describe a protein. Well, a protein contains carbon, hydrogen, and oxygen, usually nitrogen and often sulfur. It is a long chain of amino acids. Amino acids are the building blocks of proteins. And it has 4 levels of structure. The primary structure is the chain of amino acids. The secondary structure is either a helical structure, like a spiral shape, or it's either a pleated structure, like if you've ever made a paper fan before, it's a pleated sheet structure like that. And then the tertiary structure is the final 3-dimensional structure. And the quaternary structure is combining several tertiary structured proteins together. So it's a combination of several tertiary structures linked together. And boiling a protein can denature the protein and affect its properties by completely destroying the properties that it has. Okay, now our last group of macromolecules are called nucleic acids. They are composed of, you might expect by now, carbon, hydrogen, oxygen, and like proteins, nitrogen. But nucleic acids also have phosphorus, whereas proteins typically have as their 5th element is sulfur. What nucleic acids do is they store and process information inside cells. DNA is the main class of nucleic acid. It's also known as deoxyribonucleic acid, and it contains our genetic code for our genes, for our traits. RNA, or ribonucleic acid, is used in producing proteins that give us our traits. So the building blocks of nucleic acids are called nucleotides. And a nucleotide has three main parts. It has a sugar, and it's a pentose sugar. In other words, it has it's like a pentagon, has a five-sided shape. And then a phosphate group, kind of like our phospholipids that are found in the plasma membrane. And then a nitrogenous base, which includes, could be four possible different bases. There's adenine, thymine, guanine, and cytosine. And those different bases, adenine, which has two rings, guanine, which also has two rings, cytosine, and thymine, the different combinations of those bases is what determines the code for our traits, our genetic code. Now, the only difference in RNA is that DNA contains thymine, RNA contains uracil. Uracil replaces thymine in RNA. The structure of DNA and RNA. RNA is simpler. It's a single chain of nucleotides, whereas DNA is a double chain that is also shaped into a helix, helical structure. DNA has a twisted ladder or double helix shape. And this shape is held together by hydrogen bonding between the bases, between the nitrogenous bases. We also know this, if we know the sequence of one side of the DNA, then the other sequence we know because of complementary base pairing. We know that adenine always bonds with thymine and vice versa, and cytosine only binds with guanine and vice versa. Describe a nucleic acid. Well, it is a chain of nucleotides. And it contains our DNA code. It's involved in producing proteins that determine our traits. A large organic molecule composed of ribose sugars, nitrogenous bases, and phosphate groups is which kind of nucleic acid? RNA. Because if it was DNA, the sugar would be, sorry, that looks terrible. If it was DNA, the sugar would be deoxyribose and not ribose. The fact that the sugar is ribose means that it is RNA. All right, we have another compound that's very important. It's ATP. Our body uses ATP for energy. ATP, our cells use ATP for energy, sorry. ATP is a combination of adenine, just like you saw in the DNA, ribose, that pentose sugar, like you saw in DNA, and then three phosphate groups. It's called adenosine triphosphate. And our body obtains energy by breaking that phosphate bond and reducing the ATP to ADP, adenosine diphosphate. So the way that our body stores energy or makes ATP so that we can use the ATP for energy is it adds a phosphate to ADP. And the way that it uses the energy in the ATP is to break it down. It breaks it down into ADP plus a phosphate group. So describe ATP, it's adenosine triphosphate. It is adenosine combined with ribose sugar and three phosphate groups. And when ATP is hydrolyzed, the hydrolysis of ATP results in ADP and phosphate. So in other words, it would be adenosine and ribose bonded with only two phosphate groups. That would be your ADP, contains 2 phosphate groups, diphosphate. And then the extra phosphate has been removed because when that phosphate is removed from ATP, that is how the energy is released from the ATP. Biochemical building blocks are components of cells. Carbohydrates provide energy and form specialized compounds. Lipids form plasma membranes and also hormones. Proteins can act as enzymes. They can act as support proteins, and they can also form hormones. Nucleic acids contain the genetic material for our cells and direct the cellular activity and synthesis of all proteins. Identify 6 elements common to organic compounds, and you can remember this by the acronym CHNOPS: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And the building blocks that are the components of cells are carbohydrates, lipids, proteins, and nucleic acids. And if you want to talk about the building blocks of the larger molecules, building blocks of polysaccharides are monosaccharides. The building blocks of nucleic acids are nucleotides. The building blocks of lipids are fatty acids and glycerol. The building blocks of proteins are amino acids.